Good morning everyone. Myself Dr.Shweta Humbarwadi from Department of Microbiology, CKThakur College, New Panvel. And today we are going to study about the types of media for microorganisms. So basically on the physical state of media, three types are there. First one is solid media, semi-solid media and liquid media. In these three media, these are all supportive media for the growth of organism. The difference is, basic difference between these three is the percentage of agar. In solid media, it is 1.5 to 2 or 3%, and which provides the surface for the isolation of colony isolation and morphology studies. The second one is semi-solid media. This is used for in some biochemical test which contains 0.2 to 0.5% agar, which is again used for the testing of motility and some biochemical test. The third media is liquid media, which is totally liquid in form, where agar is not present, and it is again used for some the growth of the organisms as well as for the biochemical studies. And then again, on the basis of chemical composition of media, there are two types: defined media and complex media. In defined media, the composition of each and every ingredient is known, which can be regenerated at the same percentage. And in complex media, the components are different, their composition is also different, and you cannot generate the similar type of media the next time. Defined media, this is also called as synthetic or chemically defined media, and this is precisely known its chemical composition. So every ingredient is here measured accurately and added into the media. The same media we can again reproduce. This type of media is essential for nutritional studies, metabolic research, and understanding specific growth requirement of microorganisms. The example of this defined media is minimal medium, where we can add the glucose or salts according to our need for the growth of E. coli. The next one is complex media. Yes, as I earlier told you, the complex media, which is again made up of different types of ingredients. The composition of ingredients is different, so it may contain the beef extract, yeast extract, and peptone digest. The components of these are vary according to the content, and these undefined media supports a wide range of microorganisms because they provide diverse amino acids, vitamins, and growth factors. So while the exact chemical composition is unknown, the complex media is ideal for the general cultivation and routine laboratory work. The example of these are like blood agar or McConkey's agar, nutrient agar, etc. Then comes the selective and differential media. This type of media we can use for the growth of organism as well as for the identification of organism. If you are interested in the specific organism, then we can use such type of selective media. So this selective media contains agents that inhibit the growth of unwanted microorganisms. Only interested organisms will grow on this selective media. And differential media is used for the differentiating between the organisms, and this differentiation is based on their metabolic activities. So here are different again examples of the selective and differential media. One of the example is McConkey's agar or McConkey's broth. So here we can see the growth of organism on McConkey's agar. The McConkey's agar basically contains sugar, lactose, and indicator neutral red. So the organisms which are lactose fermenting, those organisms shows yellow color colony because of the pH indicator neutral red, and non-lactose fermenting organism will non-lactose will form yellowish color, and lactose fermenting organism will form pink colored colonies over the media. So we can differentiate these organism in lactose and non-lactose fermenting. The next example is again blood agar. This blood agar is again used for the identification of hemolytic pattern. So what is hemolysis? Hemolysis is the breakdown of RBCs into the blood. So for pathogenic culture study, this blood agar is frequently used. So on this blood agar, we can differentiate the organism or their pattern of hemolysis in three ways, that is alpha hemolysis, beta hemolysis, and gamma hemolysis. Alpha hemolysis, where the RBCs are partially breakdown and we can observe the metallic clear zone around the colonies. In beta hemolysis, we can observe the clear zone around the colonies, which is there is a completely breakdown of the RBCs. And in gamma hemolysis, if the organisms are not hemolytic, or they are not going to break down the RBCs, such type of organisms or their pattern is called as gamma hemolysis. Then comes mannitol salt agar. This mannitol salt agar is again selective for the halophilic organisms because it contains high salt. And again it also differentiate between the mannitol fermenting organisms and non-mannitol fermenting organisms. So those mannitol fermenting will form the yellowish color colonies because of again the pH indicator phenol red in this, and non-mannitol fermenting colonies will shows the pink colored colonies over the media. So here again we can differentiate the Staphylococcus species, where the Staphylococcus aureus is mannitol fermenting, which will show you yellow color colonies, and the non-mannitol fermenting Staphylococcus epidermidis will observe in pink color colonies. So these are the basic types of the medias. Now what are the applications of these media? So these culture media are used for microorganisms, serve diverse applications across multiple fields, like in clinical diagnostics, which relies on selective media to identify pathogens quickly. In food microbiology, it uses enriched media to detect the contamination in the food. In environmental microbiology also we can use the environmental testing, which employs the differential media to monitor water and soil quality. And in research, laboratories utilize the specialized media for studying microbial physiology and genetics. So what are the key takeaways from this topic? Choose the media based on organism requirement. First of all, decide which type of organism you are going to isolate and study further. So accordingly we have to use the selective media and then the differential media. These types of media are used for the identification of organism on the basis of their growth pattern, culture pattern, etc. Then S, align the media selection with research objectives. What are the objectives of research, where you are going to utilize it, and accordingly you have to use the media, you have to prepare the media. So this is all about the types of media and their applications. Thank you very much.